Welcome, everybody. It's 10 o'clock a.m. in Baltimore. Welcome to the 40th anniversary celebration of the Johns Hopkins Center for Alternatives to Animal Testing, or CAAT. I'll be your moderator today. I'm Martin Stevens, and I've had the privilege of working at CAAT for the past decade. And I think it's fair to say I've been part of the extended family of CAAT for over 30 years. So we're going to devote as much time as we can today to our speakers. So I'm going to be brief with these introductory comments and as well as my introduction of speakers. So my apologies in advance to the speakers for butchering their biographical sketches that they may have sent or misremembering some things about their past. I think we're all set on technical stuff. Camilla Januario will be behind the scenes, making sure everything goes smoothly. And I think we're all set with our speakers. We plan on recording today's webinar and uploading it to YouTube. So you'll notice that you've been asked to hit the right icon when you join, that you have got it. And understand that we're recording today. We'll also use the chat function for you to put random comments or recollections about your interactions with CAAT over the last 40 years. If you have an anecdote, a particular story you want to put in there, please do. Okay, our first set of speakers includes Andrew Rowan and Alan Goldberg. Andrew is the founder and director of Animal Wellbeing International. He's worked at the Fund for the Replacement of Animal Experiments, Tufts University, and the Humane Society of the United States, or HSUS. He's the recipient of numerous awards, including the HSUS's Russell and Birch Award. Alan Goldberg is professor of toxicology at Johns Hopkins University. He's the founding director of CAAT. We would not be here if not for Alan, and we'll be hearing much more about his achievements throughout today's event. Andrew, Alan?
Thank you, Marty. I'm going to just share my screen quickly. I've got a few slides that I wanted to share, and it takes, Camilla has told me that it takes a few moments to come up. So let me just begin by saying that Alan and I sort of both had sort of backgrounds in the beginnings of CAAT, and so we've divided our presentation up. I'm going to talk a bit more about what was going on in the larger environment back in 1980-81, and Alan's going to talk about the actual formation of CAAT. So first of all, congratulations on 40 years. It's been a phenomenal journey, and I think that everybody can be very proud of what has been achieved. So in 1980, in the United States, animal protection movement spending was around about $6 per capita. That's in 2020 dollars. And there were perhaps 2,000 organizations, animal organizations altogether in the country. By comparison, today, the spending is double that in inflation adjusted dollars, a total of 3.7 billion, mainly on dog and cat issues and advocacy. There are 22,000 animal organizations today. And animal research and testing is no longer a major focus for the movement, but it is still present. And now you've got Unilever, P&G, and L'Oreal working with animal advocates. That was not the case back in 1980. Just to place a little bit more context, so the campaign against the cosmetic companies started in the UK, basically. It was launched in part by groups now, Animal Aid, some of the AV groups. Frame was part of that. We had a brochure called What Price Vanity that helped, in fact, found Animal Aid. And there was some activity in the United States at the time with the International Society for Animal Rights producing a fairly comprehensive document, about a 30-page report on animal testing in the cosmetic industry. But there really wasn't a great deal of focus at that time. That focus came when Henry Spira decided to address the Drays test, started the campaign to abolish the Drays test, the coalition to abolish the Drays test. And he then targeted Revlon. So the CTFA, as it was then known, Cosmetic, Toiletry and Fragrance Association, initially ridiculed the efforts of animal advocates. I remember talking to Norm Estrin, who was their chief scientist in 1980, and he pointed out with laughter that the animal protesters at the CTFA were a block down the road in front of another building, not in front of the CTFA building. So, I mean, that was a general attitude about the issue back in 1979, 1980. And then there was this focus on Revlon through 1980, basically pushed by Henry Spira and his coalition. And in December, Revlon announced $750,000 to find an alternative to the Dreys, a grant to the Rockefeller University. This is probably familiar to all of you. But what people may not know was that that announcement was made in the middle of the CTFA's Science and Regulatory meeting in Florida, which I was speaking at the time. Not a warm and fuzzy welcome, I remember. And Revlon was peeved because they had been taking it on the nose for the industry, as it were. And so they invited all the other companies to join the effort to find an alternative to the Dreys test. That was, a major wake-up call for the industry. And what happened then was the CTFA, with Avon leading the way, put together a fund that then ended up at Hopkins. I would say that NIH at the time was very much resistant to this whole notion of alternatives. They talked about complementary methods instead. And in fact, they did not actually use the word alternatives unless they were responding to a specific legislative language until this century. So it was a tremendous resistance to this just very notion of this particular idea over the years. At the time, animal advocates were viewed as misguided at best and misanthropic and emotional idiots at worst, and a fair number of people felt that way. The corporations that initially got involved basically got involved at first for PR purposes, although that changed fairly quickly. Within 3 or 4 years, corporations are saying, wait a minute, we think there's some real sort of heft here, some real issues here that we can address. And CAAT's establishment at Hopkins was a major change in the whole landscape. And I have to basically applaud D.A. Henderson for having the vision to grasp that particular nettle. And one final anecdote, shortly after CAAT was established, I was invited to go to Bristol Myers Squibb. I was talking to their senior VP for public relations. And he sort of handed me a letter he had just received from Hopkins, the president of Hopkins, asking Bristol Myers to help them fight this alternatives issue. As it happened, of course, CAAT had already been established, and the vice president sort of said, do these people not talk to each other? And of course, we all know that in universities, the left hand often doesn't know what the right hand is doing. But that was one final little anecdote that I remember. Hand it over to Alan now for his thoughts. Let me see, I got to stop screen sharing.
Yeah, thank you very much, Andrew. They brought back a lot of great memories. So you mentioned D.A. Henderson. D.A. Henderson, for everyone else, was the man that eliminated smallpox, the only disease ever eliminated from the world. But he became the dean. And on a plane ride, he was sitting next to a man by the name of Bob Worsfold, who happened to be also the chair of our advisory board for the school. And Bob was the president of a cosmetic company. And he approached DA with sort of the question, could you help us deal with the animal activists and help us get out of animal testing? Well, it ended up that at that time, I had just published a paper with a man by the name of Neville Brooks using tissue culture to look at a specific function that we could not do in animal studies. DA was aware of that and contacted my chair and me to see if we could set up something to help the cosmetic industry deal with non-animal methodology. And it was $1 million. That was a rather large sum. At the time, the average NIH grant was $40,000. So this yearly sum was 10 times the amount of the average NIH grant. The internal discussion that took place, and there were actually 4 of us involved in that: D.A. Henderson, Gareth Green, Frank Lowe, and myself. And when we talked about $1 million coming in for 3 years, we all felt that that was going to be the length of the entire experiment. Well, we know we were wrong. 40 years later, we're up and running and actually growing on a regular basis. So the first aspect was to get a program in place. The second thing that happened was we decided we needed to have an advisory board that was really independent and could provide advice and mentorship. So that first advisory board, we were founded in September 21st. Today is the day, in 1981. And in February of 82, we had our first board meeting. It is something that has I have vivid recollections of for many reasons. Number one, most of the people in the room did not know each other, which was very unusual to start with. And two, some of my academic colleagues were more animal welfare friendly than even those that were from the animal welfare world. They were very, very serious. And I think those kinds of things have stayed with the center in terms of the animal welfare components and quality science. So if anybody has ever written an NIH grant, you know that you write a yearly progress report and nobody ever reads it. In this case, we had to provide quarterly progress reports. So I sent in the first progress report. I believe it went in on a Friday. Monday morning, first thing, I had a call, the secretary got a call from the scientist at CTFA, Norm Estrin, to speak with me. I wasn't immediately available, and she gave me the message when I was. And my comment was sort of like, "Hey, here they are interfering already. We're only 3 months into this." But I called Norm back and said, "How can I help you?" And he said, "I want to thank you for this very informative report." Number one, he read it, which was unique to me. And number two, he was going to be helpful. It was sort of a very critical point of change between my thinking in terms of what the sponsors were going to want and what we could achieve. So many of you know the rest of the issues. Hopkins was both very friendly towards the center and wanted to get rid of it at the same time. I mean, it was clear that there were people that thought we were doing the worst thing possible. We had capitulated to the worst forces that there could be. And yet at the school, because I did have, something just went crazy. Because I did have, I don't know if anybody can see it or not. Okay, we're back. We did, at the school, we had good support. And that was one of the reasons. The second reason, I think, was I was a respected scientist at the time. And clearly that made a difference. Why would I go into something like this? So over the years, we created many programs. And I am remarkably happy to say that Thomas, when he came in about 12 years ago now, maintained and expanded those programs, but also started several new programs. And I think the rest of today, we're going to hear about all of those programs, the early ones and the later ones, and where they've gone. The center has been absolutely remarkable on a world scale. We are known all over, and the activities are clearly dependent on the people that are on this call that were with us at the early stages. Many have passed. In fact, the three I mentioned that were Frank Lowe, D.A. Henderson, and Gareth Green have all died since the founding of the center. So I look forward to hearing as we go forward today with each of the different programs and activities. So thank you.
Thank you, Alan. Thank you, Andrew. That was a very nice set of introductory comments. So now we'll get into the heart of the program and talk about one of the efforts that Alan alluded to, that he had started a number of programs in the early years, and this will be one of them on developmental neurotoxicity. So we'll be hearing from Helena Hogberg and Tim Shafer. Helena is Deputy Director of CAAT. She's active in CAAT's lab, working on developmental neurotoxicity and other issues using our mini brain model. Tim Shafer has been a researcher at the US EPA for 30 years. During the last 15 years, his work is focused on the development of in vitro NAMs, the non-animal methods, for developmental neurotoxicity hazard identification. Helena, Tim?
Thank you, Marty. Yes, so happy birthday, CAAT, 40 years. I have not been here 40 years, but almost 12. So Tim and I are going to talk about the developmental neurotoxicity program. So before I go into developmental neurotoxicity, I just want to highlight the TestSmart program, and you can see these three individuals here, Sydney Green, Joanne Surlo, and Alan Goldberg. They were very much involved in the establishment of this TestSmart program, which was to develop a more humane and efficient approach to risk assessment by bringing together diverse stakeholder groups from around the world. And the first TestSmart workshop took place in 1999. And after that workshop, the EPA actually announced that it had decreased its estimates for animal use by 80%. So then how did this TestSmart lead into developmental neurotoxicity TestSmart? Well, these three individuals here, Pam Lane, Alan Goldberg, and Kevin Crofton, were very much involved in this. And Alan couldn't remember what year, but I estimate that it must be after 2000 and before 2005. When these three people met in Aspen, Colorado for dinner, and they talked about DNT and how to set up the first meeting. And this is a quote from Alan that he said, "I chose DNT as a topic for TestSmart as there were no specific requirements for DNT. So we did not have to match against an in vivo approach." So that's why how it all started. And then here again, Pam and Alan, and together with Paul and Ellen Silbergirl, they established or discussed more into details how such an approach would look like, the DNT Smart. And this is another quote from Alan saying, "We held a European meeting to convince whoever was the responsible person from GRC that DNT was important." So they had the first workshop in ECVAM in 2005. And this is the report from there. So here you can see that some of the responsible people from the GRC from this workshop. Two of the main key players from the GRC were actually Sandra Coecke and Anna Price. And they are still, at least Anna is very much involved in the DNT still, and Sandra is very much involved in the alternatives. But then we have two other people here, Kevin Crofton and Bill Mundy from USEPA, that also had a very big role in the establishment of this, of course, together with Alan. So this workshop followed by some international conferences, DNT Smart, or TestSmart DNT, so 1 to 4 here, where CAAT was the main organizer of 3 of them, but in very close collaboration with the USEPA and the GRC. And the goal for these programs was to identify alternative DNT models, and then to develop a process for validating these test methods, and in the end to reduce, refine, and replace animals in DNT. So here was some of the pictures that I found from the DNT, the first meeting. So you can see here Marty Stevens and Alan Goldberg that was presenting at that meeting. And then at the DNT2, I actually took 2 slides from the presentations that were presented at that time. And interestingly now, this was from Tim Shafer, the first one on the top there, and the lower one was from myself. And the reason I picked these 2 was because at that time, in 2008, when DNT2 took place, these 2 test methods had screened the most chemicals. And as you can see here, it's like 10 to 15 chemicals, and that was a lot of chemicals at that time. Going fast forward, Tim will tell you a little bit what's happening now and what have happened during the years. But so of course, these workshops or these conferences brought a lot of discussions together with all different stakeholders, and a lot of reports came out of this and a lot of progress. But one of the highlights I would say is this paper here, which we often call the Crofton paper, which is based on the discussions on these conferences. And it is actually a description on how to develop an alternative for developmental neurotoxicity. CAAT also had a laboratory. Here you can see an old picture, this old long time ago when CAAT started his in vitro toxicology laboratory, and you can see Joanne there, for example. Then the activities in the lab was reduced over the times. And in 2009, 2010, when Thomas came back, came as the director, we actually re-established the lab. And here you can see myself and Erwin van Vliet, which was the 2 first postdocs. And of course, our work was done on developmental neurotoxicity, and in the end led to the development of the human mini brain models that we were using today. So then together with CAAT Europe, we organized several workshops, and here are 2 of them shown. And the main outcomes from those were these 2 publications. One came up with a list of reference compounds for DNT, and the other one, I would say maybe one of the major achievements also, is the recommendation of test readiness criteria for NAMs for developmental neurotoxicity. So DNT5 was going to take place in 2020. However, due to pandemic, it has been postponed, but it's still going to happen. Another achievement, big achievement, was the establishment of the International Stakeholder Network, ISNET, which is different stakeholders brought together, especially talking about the regulatory approach using these alternatives for a test roadmap for regulatory purposes. And here we started to talk about using the adverse outcome pathways for developmental neurotoxicity, as well as the integrating testing strategies. And by that, I want to hand over to Tim Shafer to continue, and I will still do the slides here. So I go for,
okay.
Sorry, now everything is going really fast.
I can't talk that fast. Well, hello, everybody, and happy birthday to CAAT. It's nice to be here today, and I'll send also greetings from Bill Mundy. I talked to Bill yesterday. He's doing well, and sends his best wishes to everyone. So I'm going to pick up on the legislative aspect. So if you could hit the slide one time, Helena. So going back to, wow, it's really fast.
Let me stop sharing and restart this because it's
okay.
Good idea.
Or if you have trouble, Helen, I've got my presentation up. I could share it too. We'll try one more time with you though.
You're muted, Helena. I would suggest you.
Yes, Tim, can you share yours really quick? Because I think that was
yeah,
just
hang on a
continue.
second. Let me
Sorry about that, guys. It's sometimes it's keeping the recordings and it just popping up.
okay. Okay, so I'm going to pick up with the legislative aspect, as I said, and I have to turn to my other screen here, so you'll see me facing the other direction. But I think I've picked the wrong slide. Hang on one second. Okay. Okay, here we are. Going back to, as early as 1996, some important legislative developments have been happening in the last 15 years or 25 years now. The Food Quality Protection Act that was passed, it was an amendment to FIFRA, to FIFRA in the United States, and it specifically said that the developing organism has to be protected, and that we have to consider that in our risk assessments, and we have to use the best available science, which includes in vitro data, when we do that. And then following that, about 10 years later, the REACH legislation was passed in Europe, and that also encouraged the use of non-animal or in vitro approaches to have data to make decisions about chemicals. The United States was a little bit slow in responding to that, but we did eventually get the job done and pass similar legislation in terms of the Lautenberg Amendment to TSCA, which covers all chemicals in the United States. And that amendment specifically said that the EPA will use alternative approaches whenever they are possible and feasible. And so I think these are three really important pieces of legislation that were influenced by the work of CAAT and other groups that were advocating for alternative approaches for toxicity testing. And then so the Lautenberg Amendment was passed in June of 2016. And then in October of 2016, there was a meeting that was held sponsored by the OECD and EFSA in Brussels that focused on the use of non-animal approaches for developmental neurotoxicity. And this is really, I think, a seminal meeting because there were a number of important outcomes that flowed from that meeting. One of the things that happened at that meeting was Kevin Crofton specifically called out the field. I'm going to see if I can move there. Specifically called out the field for, although we'd developed all these assays, and these assays are listed in the columns across here, these were all various different types of DNT in vitro assays or using alternative species such as zebrafish or planaria. We'd really tested very few chemicals, especially those that were relevant for developmental neurotoxicity, there's a chemical space above this purple dotted line. And so Kevin really hammered on this point, and he said, we need to test more chemicals in these assays to better understand how they work. So my computer is very slow today. Apologize. So again, going back to that workshop, one of the outcomes of that workshop was that the OECD through EFSA issued a grant for people to specifically test more chemicals in these DNT assays. And that grant was awarded to Marcel Leist and to Ellen Fritche. And then they approached myself at the EPA and said, let's work together and try and test as many chemicals as we can in the assays that we have. And so we worked to develop a list of chemicals, looking at what had already been tested, for example, in different assays versus what had not been tested yet, to come up with a list of about 120 different chemicals that we could test. At the same time, the OECD formed this in 2017, formed this expert group on developmental neurotoxicity. And the goal of this expert group is to develop a guidance document for the use of in vitro DNT assays using the integrated assessment and testing approach, or the IATA approach. And so that is ongoing currently. Also, the National Toxicology Program had distributed a set of chemicals to a number of investigators, and there were about 30 developmentally neurotoxic chemicals in this set, and they held a workshop to focus on the outcome of the testing for those chemicals in September of 2017. So 2016, 2017, a lot was happening. What came out of this meeting in 2016 was also the development of a test battery, and the proposal that these key processes would be tested, and that these were the assays that we could use to test these processes. And this was the first proposed in vitro test battery. And I want to point out that I can't put all these references on timeline, but clearly these test assays in the battery were strongly influenced by the previous, DNT TestSmart meetings. All of them were by people who attended those meetings or their principal investigators and incorporated the ideals that were set forward and developed in those DNT TestSmart meetings that were sponsored by CAAT. And then also I wanted to point out that in 2019, NTP released their DNT diver tool, which visualizes the information in the assays that people submitted data to the NTP for their testing program. And this is a nice visualization tool if you haven't used it. So in September of 2020, we held a scientific advisory panel under FIFRA to review the use of new approach methodologies for DNT. And you can see we had data on a number of different organophosphate pesticides that were tested in the EPA assays. One of the outcomes of this meeting was that the scientific advisory panel in their report stated that they did agree with the EPA that these assays could be used as a part of a weight of evidence approach for developmental neurotoxicity testing. And then following that in October, the report from Marcel and Ellen's work testing the DNT chemicals was released. And you can see here now that we have data on about 100 chemicals. And so we're starting to realize the goal that Kevin had laid out of testing more chemicals. And I'll note that the EPA data will be released through ToxCast with its update next summer. And so we're trying to incorporate as much of this data from Marcel and Ellen's labs as we can. We don't know if we'll be able to make that public yet, but I know they're also developing a database to make that public. So there will be a lot more data on these assays, and that will allow us to better interpret the data coming from them. What's happening now is that the National Toxicology Program just distributed about 115 chemicals to my lab, Ellen's lab, and Marcel's lab, as well as BioBide, which uses zebrafish for testing in these DNT assays. And so that data should start to come in next spring and next summer. And then this summer, and many people are not aware of this yet, but the EPA used data from a couple of the DNT NAMs as support for waiving a guideline DNT study. The in vitro data and the other available data indicated that there would be no added benefit of conducting a DNT guideline study for some chemicals that were proposed for registration. And so that was waived. And to my knowledge, this is the first use of DNT NAMs data in a regulatory decision by the EPA. And so I put a star by that. And so we're actually now using this data to help guide decision-making. The expert group on DNT, the guidance document has gone out for its first round of review. And so the partners are providing feedback. I think the last of it is due by this Friday. We'll then be revising that document, and it will be moving forward through this next year. And so as Helena mentioned, we hope to see that DNT5 is in the future. And I think there are lots of things that can be discussed. I think that we're right at the beginning of DNT NAMs. And I hope that as we move forward, we can improve these NAMs, we can get better coverage of the biological space, and that in the future tiered testing approaches will be developed, among other things. And so I really think that the impact that CAAT has made in this field has been tremendous, and I hope that I've highlighted a couple of the reasons why. And so again, happy birthday to CAAT, and thanks for allowing me to be part of this celebration.
Thank you so much, Tim and Helena. It really does illustrate CAAT's contribution to this field nicely. And of course, for those new to the field, NAMs are new approach methodologies. Okay, we will move on. Since this is a celebration and not a scientific symposium, we won't be taking questions per se, but maybe there'll be time at the end to have a discussion about some of the more technical issues or other issues. And I would, again, welcome people to put celebratory comments in the chat box if they would like. Okay, moving right along to yet another program of CAAT over the years, a newer one that Thomas inaugurated at CAAT when he came, evidence-based toxicology. So Sebastian Hoffmann and Daniele Wikoff will be talking about CAAT's effort, its impact, and the wider field of evidence-based toxicology. Sebastian Hoffmann is a co-founder and staff scientist of the Evidence-Based Toxicology Collaboration, or the EBTC as we call it. He did his PhD research on evidence-based toxicology under Thomas Hartung, and worked with Thomas at ECVAM for many years, the European Center for the Validation of Alternative Methods. And Daniele Wikoff is the Director of Tox Strategies Health Science Practice and is the chair of the Scientific Advisory Council of the EBTC. She specializes in the use of evidence-based methods in support of hazard and risk assessment applications. Sebastian and Daniele, the floor is yours.
Thank you, Marty. I'll go first. I'm trying to share my screen. Can you see my slide?
Yes.
Great. So first of all, happy birthday from Europe here to CAAT to the 30th birthday. I'm quite honored to be part of that celebration here today and part of the program. And I'll briefly present a little bit one of the more recent childs of CAAT, the evidence-based toxicology collaboration. In fact, we're turning 10, or we turned 10 this year, so we still can behave a little bit more childish probably than CAAT can afford. We have a brief presentation, just a handful of slides, and we then chose to have more a back and forth a little bit, or Daniele picking some of the highlights of the last 10 years of the EBTC and its program. For those of you who don't know the EBTC, our aim is to bring together the international toxicology community to facilitate the integration of scientific evidence into regulatory, environmental, and public health decision-making. In a little bit in more detail, the objectives of our work are to promote high quality, high impact research in toxicology and environmental health. And here on the slide, you see 4 themes. All our activities and projects can be assigned to one of those themes. I'm not going into any great detail here, but the themes are raising research standards. So we're concerned about improving the methodology, the research methodology used, but also, for example, to improve the reporting of research so that it then can be better used when it comes to integrating it with other information. Another theme is making sense of evidence, and under that, all our review types can be subsumed. We have conducted a couple of systematic reviews, scoping reviews, and other types of evidence-based evidence integration. Another theme is we're aiming at improving access to research. So that has, of course, the notion of making research open access, but also structuring research in a way that it's easily accessible, that you can retrieve data from papers more easily than it's the case today with unwieldy PDFs, for example. And just embarking on the 4th theme you see on the right, that's advocating evidence-based decision-making, but that's a new addition to the program, or it's going to be a new addition to the program. So very briefly, who's running the project so that you have faces to us. Our director, Katja, who couldn't make it today, is steering the boat, and Paul and I on the left and right hand side do a lot of project work and support her. This is already my last slide, but it will be a very busy one. So I try to do very similar things like Tim and Helena did, squeeze in some highlights of the last 10 years into one slide only. So there was some initial work starting in the early 2000s, and here's just the highlight of that period. In 2007, there was the first international forum towards an evidence-based toxicology. There was a forum held in Italy, organized still at the time when Thomas Hartung was head of ECVAM, and I was working with him there. Then there was a small break, and in 2011, so 10 years ago, the EBTC was formally kicked off at the SOT. So that was probably in March, and later that year in June, also at the Eurotox held that year in Stockholm, Sweden. Then in order to get some visibility and some attention, we focused in the early years on organizing workshops. So one in 2012 was held at the Research Triangle Park, which is the one on top. So evidence-based toxicology for the 21st century, opportunities and challenges. And as you can derive from the title, there was around that NIS report, 21st century toxicology. And 2 years later, we had a workshop a bit more focused that was primarily on the emergence of systematic review and related methodology. And that was held in Baltimore. At the same time, we tried to reach out to the larger toxicology community and had symposia at the International Congress of Toxicology in Seoul, 2013, and also 3 years later in Mexico at the ICT. But already in those early years, we grew a little bit and felt that we need kind of more structure, more organization around this growing activity. So we sat together with Marty and some others and created some bylaws for the EBTC, established a board of trustees and also a scientific advisory committee. And that was 6 years ago. After that, we then managed to get our first scientific papers published on top. Also, Marty was here heavily involved with the scoping review on guidance of assessing the methodological and reporting quality of toxicologically relevant studies. That's a quite well-cited paper, and it was a contribution to a special issue by Environment International. Then another highlight publication was what we call their systematic review primer, in which we brought some stakeholders together and we tried to apply the systematic review to toxicological questions, highlighting the challenges and problems in that adaptation. Then again, we had a period in the last years where we had a lot of workshops. I think this was one of the highlights. There was a big workshop jointly with the European Food Safety Authority, that was held in Portugal on evidence integration. And we followed up with two a little bit smaller workshops, one co-hosted by GREAT and the other again by EFSA, where we focused on integrating mechanistic evidence into systematic review and in chemical assessment frameworks. Very recently, we finally published the systematic reviews we conducted ourselves. The first one was on the TOX21, we call it, was on the performance of preclinical models in predicting drug-induced liver injury in humans. And the second one is a systematic review of the zebrafish embryo toxicity assay and compared it against the mammalian prenatal developmental toxicity assays. So applying systematic review to compare two toxicological test methods. And here's a very brief outlook before I hand over to Daniele. Next week, we are having a continued education course at Eurotox on Monday morning, a 4-hour course on systematic review in the field. And with that, I'd like to hand over to Daniele, maybe to chime in and give her view on where she sees the EBTC.
Sure, thanks, Sebastian. Well, I haven't been involved for this entire timeline, but I came in somewhere in the middle, and I can say from where I sit, having been involved in a number of capacities with EBTC, but also as a practitioner of systematic review and evidence-based techniques in my day-to-day, I think that the organization as a whole has really been successful in providing a platform for discussion and truly for collaboration. When you and I look at this slide, I would certainly agree that these are only highlights in main events, because one thing that I think happened from these events, and particularly those on the right, when more people started to get involved, is that the discussions really continued, and more sessions or workshops at other meetings relating to these methods have really been a result of some of these sparks that the EBTC has really helped to generate. Also, the collaborations and the relationships that have been built through these workshops, et cetera, have been very successful. I also think that the right side of the slide, highlighting the emphasis on mechanistic data. What is mechanistic data? Well, in risk assessment and hazard assessment, this is really important to us, and it usually relates to events leading up to an adverse outcome, of course. But the challenge in risk assessment is understanding how to put all that great information together. We've just heard a lot about the different methods and assays that are being developed for alternatives. But how do we put all of that together to inform decision making? And it's very clear that evaluating and integrating mechanistic data in an evidence-based manner is a huge challenge. And so the EBTC has really supported collaborations, discussions, et cetera, on everything from methods to different applications, hazard, AOPs, mode of action, and has been successful in bringing together people that have been thinking about this from a number of different capacities. And so we're really looking forward to the Eurotox session. It also has a high focus on the mechanistic or alternatives data and how to think about that in a very applied manner for risk assessment. I would also say through this combined, all of this, I guess, success as time has gone on and more people are interested, more people are attending, asking questions, thinking about projects, conducting projects, trying to publish. It's been very evident that the organization it's time for an update or a little bit of a refresh in terms of the structure, which will allow this to continue, the organization to continue to grow, and also to facilitate more and more scientific investigations and projects that aren't limited to the 3 people on a couple of the slides before. So we're really looking forward to what the updated vision and restructuring will bring, and the opportunities that will provide for the scientific community as a whole. Sebastian,
Great.
anything to? Yep.
Is there more, Daniele or Sebastian?
No, that was my spiel.
No, that was great. And that'll help us get a little more back on time. We're a little bit behind, but I don't think that'll be a problem. Thank you so much for illuminating what many people feel is a challenging area to wrap their minds around. I think that was quite clear. Okay, so let's move on. The next segment will be on good cell culture practice, and we'll be hearing from Marcel Leist and Sandra Coecke. Marcel is the founding director of CAAT Europe, which is based at Constance University, where he's a professor. Sandra Coecke is a senior scientist at European Commission Joint Research Center. She's been managing and leading scientific teams since the 90s, first in the pharmaceutical industry, and later joining the Joint Research Centers at ECVAM, the European Center for the Validation of Alternative Methods. And I'll turn it over to Marcel and Sandra.
Well, thank you, Marty, for the nice introduction, and welcome everybody to the birthday party of CAAT. So don't run away, don't yawn, like if you have been working with cell cultures and you think you know everything about it. So there's 3 important things in cell culture. So that was the Nobel Prize in '54 for the cell cultures, which actually was the resolution of the big pandemic of polio, or one big contribution. Then, of course, the Nobel Prize 2006 for the stem cells, and of course, good cell culture practice. And my co-host Sandra is really the ideal person to talk about this and give you a little bit more of a historical introduction. So Sandra, please join me.
Okay, thank you a lot, Marcel. And again, happy birthday to everybody. It's so joyful to see a lot of the faces and friends in this area. And I have some liaisons with the previous talks, and I will show them why we, let's say, like Ellen said, in this field, it's good to know each other and work together. So let's start saying what we mean by GCCP. So it started all quite early on in 1999. So we heard already about that day, 1999, from Ellen. And I'm going back. Marcel, can you go back to the slides previously? Okay. So the definition was set, and we had a meeting, and there was Thomas Harting and Gerrit Granthaler, and they really called on the scientific community to develop guidelines defining minimum standards in cell and tissue culture. And that was a GCCP. And obviously, they should encourage science. Now, Ellen said already, now you can go to the next slide. Ellen Goldberg said already, what we needed to do is to, let's say, get quality science, because quality in science gives trust. And actually, this is also something we have been working on all together to, let's say, get trust in these new generation methods in that time, now new approach methods. And so we have been working on 2 areas. One is really the more formal area that everybody knew, which is working with this guideline, the GLP. So one area of thinking about how we can get trust in these new methods, we started to work on getting an advisory document at OECD on GLPs. And then the GCCPs was really targeting the research community. And in a way also what has been said by Sebastian Hoffmann, what we want to do with GCCP overall is also a bit what EBTC wants to do. We wanted to raise research standards. That doesn't mean that researchers don't work according to standards, but there are some pitfalls. And let's say all these endeavors have been done to try to point to the people where things can go wrong. And as Sebastian said very nicely, that we want to avoid that people are wasting research resources. So all this enters there. So there you go, Marcel. Marcel,
Marcel, I think you're muted.
Yeah, I have been silenced, sorry. So I think after the general timeline introduction, of course, it's important to go along the principles, at least very briefly as a reminder. And I start with the easy ones and move to the harder ones. So principle 3 is documentation requirements. It's not so difficult to understand. Then principle 5 is compliance with laws and regulations. This is, of course, also not so difficult to understand. Principle 4 is measures to avoid hazards to the individuals and the environment. This is clear, but it's already some step to think ahead. And then also something that sometimes gets forgotten or neglected is the principle that adequate education and training should be provided. Now I come to the two really heavyweight principles, and they take a lot of space in the document. This is establishment and maintenance of a sufficient understanding, this is a heavy word, of the in vitro system and of the relevant factors which could affect it. So this goes deeply into science and not just using cells, very simply. And then the second principle is assurance of the quality of all materials and methods and of their use and application in order to maintain the integrity, validity, and reproducibility of any work conducted. So if you really digest these sentences, you understand why a more extensive document is required to really explain what is meant there. And this document was set up in 2005, as Sandra explained. And of course, a lot of things have been happening since. For instance, as I mentioned, the Nobel Prize for the pluripotent stem cells, and then of course their broad application. And of course, they are still cultured cells, so the principles we have just looked at still apply to these cells. But they also bring additional requirements, especially when they're used not as simple cultures, but within microphysiological systems in complex cultures. And therefore, the idea was born to update good cell culture principles to a version 2.0, which is intended as a guidance also for many new aspects and issues. And just to briefly summarize what some of the new challenges are, of course, the induced pluripotent stem cells, they have a higher requirement for maintenance and stability, also concerning their genetic stability. Then there are many reprogramming methods that have, of course, not been considered initially in 2005. Many differentiation protocols to more than 200 different cell types. And of course, the cells have different maturity, especially when they're, for instance, used for DNT testing, as Helena has shown. And of course, they are being used in disease models, not just for COVID, but many different diseases. And the education requirements, of course, have become more complex. And having set this stage, I think Sandra will take over now with the more regulated side.
Yeah, thanks a lot, Marcel. And as I have been showing on my first timeline, remember, we have the GLPs on top. GLPs are also part of the OECD regulatory program. And we have, let's say, the science of the GCCPs on the bottom. So now strategically is to bring these two together, because obviously thinking about trust, if we have a guidance document, look what Helena before and Tim have been explaining in the DNT world, when you go to OECD, again, we have there the trust. So at a certain point, thinking about the GCCPs, like Marcel said, the first principle is a bit about, know what you work with, yeah, and the quality of the things. So we started to work on an OECD guidance document, inspiring the people at the OECD Secretariat by the GCCPs, but covering a little bit more the whole process of what a cell and tissue culture goes through. So this is the whole process cycle of when you work with cells and tissues. You start obviously with a training facility, roles and responsibilities, and the GCCPs is an integral part of that. So that endeavor obviously has created a lot of visibility on the importance of a high quality standards in science, in any kind of science, as well as for the cell and tissue cultures. Also, it should be the same for the animal studies. But we have done this endeavor, and obviously it is resonating because OECD needs acceptance of many, many member states. Now we are 38, Costa Rica joined. And obviously, like we did with the GCCP, what we want to do is think about the science, think about the quality, and put all these things together in one document and strive for harmonization at all levels, because obviously OECD is an harmonization body. So next slide. So obviously what the idea is when we think again of what we want to do with the principles, as Marcel has been saying, is know what you're working with, get relevant data out of it, and assure that you don't do work that you have to throw in the bin. So let's say applying GCCP as part of overall good in vitro method practices by everybody, researchers and everybody, suppliers, etc., by the whole global life science community, obviously is leading to more harmonization of in vitro methods and the acceptance and uptake of that. And obviously we at the European Commission Joint Research Center, we are trying to stimulate that, and we try also that we have compliance with all these principles also in our validation work. And we are working on tools to facilitate that. And so as Marcel has been saying, you know, the science is a moving target, you know, we have scientific advances. And indeed, the original GCCPs, when we were reading that, you know, they're still okay. But obviously, there was a need to a bit reflect together what would be helpful. And so we all together started the endeavor to think about the GCCP 2.0. And Helena, who has been working very hard, can now say that we have been involving a kind of stakeholder board, the SAC, to look at what we're doing with this GCCP. We have been inviting via Altex, you know, the global community to look at what we are drafting and to give in comments. So we just last Friday got in all the comments again to, let's say, do crowdsourcing and have everybody part of this endeavor to work together in this field on, let's say, high quality scientific practices, again, for getting trust in all these new approach methods using cell and tissues, including the difficult and the new ones like stem cells, organ on a chip technologies, etc. So you see here on this slide some of the faces. And Marcel, to you.
I'm just going on because we have a crisp program, a short presentation. And of course, we want to thank so many more people that have contributed here and that gave us the chance to present this. So the Good Cell Culture Practice 2.0 is a good stage, and it is there because a scientific advisory committee of more than 50 people gave more than 350 comments. They have all been incorporated into the document, and this will soon be published. And I leave the final words then to Sandra with the contributors of the GIVIP. I think, Sandra, you are silenced somehow.
Thanks, Marcel. It's great to do these dual presentations. I mean, here again, some names of people, but let's say, I think what we have been showing in also this and the previous presentation, this field is just going so fast now, but not fast enough because all the people that are involved with it. So great that we are here today and see a lot of the other things happening. So congratulations to CAAT, to Alan, and all the people that started this endeavor.
And use the chat if there are questions or the emails which are present here. Goodbye and all the best wishes to CAAT.
Thank you, Marcel. Thank you, Sandra. That was a great presentation. Clearly, this is a collaborative effort involving scores of people, and our hat is off to you folks for coordinating what must have been a challenging project. Okay, we'll move on. We're a little bit closer to being on time now. Thank you for those few extra minutes, Marcel and Sandra. we'll move on to the topic of green toxicology. Alexandra Maertens will give that presentation. We had originally hoped to involve Nick Anastas as well, but that didn't work out. So it's just Alex. And Alex has led CAAT's program on green toxicology for several years, and she has a book coming out on the topic shortly. And she also directs our Read Across program. Alex?
Hi, can everybody hear me and see my slides? I'll assume so if I don't see anybody
Yes.
frowning. Great. So Nick unfortunately couldn't make it today, but this gives me the chance to kind of tell you the origin story, a little bit of green toxicology, which really started at the Green Chemistry Conference when Nick and I both were raising our eyebrows at the quality of the toxicology that was being presented. And of course, we had to share our despair over a beer. And this kind of led to the idea of green toxicology, or having a better, higher quality toxicology component at the Green Chemistry Conference. And really at the heart of the problem was the fact that chemists and toxicologists both kind of thought they were talking about the same thing, but often they weren't. And the story that I think really illustrates this was told to me by a chemist. And she said that she had talked to the product stewards for years before she realized that when they were talking about a rat model of disease, they were talking about an actual rat who was being dosed with a chemical. She had always assumed that that was ridiculous. That's not how anybody would study this. And it was just a computational model of a rat. So that's kind of the anecdote I like to use to illustrate the gulf between how chemists perceive toxicology and the toxicology that we're actually doing. So I think green toxicology is sort of the youngest of these programs. It started in 2014 with Green Toxicology Day. And in the same year, we had the first publication from CAAT on green toxicology in Altex. And in this, we sort of talked about the fundamental principles of green toxicology, which I sort of see as pushing green toxicology towards being a 21st century science that can really participate in the green revolution that's going to be really, really important for solving environmental problems. So the considerations are pretty fundamental. We want to be able to design benign chemicals to test early and produce safe chemicals, avoid exposure, make testing more sustainable, allow early use testing, and testing that can really drive 21st century toxicology. So one of the things that's kind of a central component of this is read across and using big data to kind of drive hazard assessment. We started in 2015, what was then considered a large data set. We had 115 chemicals. And that was our first paper on skin sensitization, but this rapidly changed when we were able to sort of use the ECA data set in 2016, which was a much, much larger data set and really demonstrated that fundamentally, if you had a large enough data set, toxicity really was a structure of function. You could make pretty accurate predictions if you had enough data. And of course, since then, data sets have really grown. In 2018, computational toxicology dashboard from EPA came online, and it's only grown since then. So now we have substantially more data than we used to have. But one of the key things, it's not just enough to produce new models, it's really important to make the models useful. So one of the things that we did was assemble a large data set of chemicals that were previously not used to build skin sensitization models to assess sort of their accuracy in a real world use case. And we had a pretty good story to tell from this. A lot of the models, they were, some of them were more accurate than others. They had higher sensitivity or specificity, but fundamentally the models were performing about as well as the animal test, which is, as well as it can do. We also just recently published a paper on chemical respiratory sensitization, which is another case study where there really was no good animal model. But we did show that at least on a small data set, that if you combine two different computational models, you got about 80% accuracy, which again is about as well as you could expect the models if we don't have a gold standard. Another key component of green toxicology is really making toxicology a 21st century science and using the tools of systems toxicology. So we've had a series of papers that have demonstrated the usefulness of in vitro models and the usefulness of high throughput data such as microarray to really understand the mechanism of toxicity. We've studied both MPTP, estrogen, BPA. That's been recently published. Another key thing is also exploring the limits of in vitro models. Way back when we were working on the human toxome project, we demonstrated what I think everybody knew, but nobody had really put numbers on, that MCF7 cells were extremely unstable cells and very, very difficult to use for hazard assessment. And we recently published a paper, again, using bioinformatics techniques to really illuminate some key differences between MCF7 cells and human breast cancer tissue. We've also demonstrated that it's important to kind of use new technologies cautiously. One of the key ideas of green toxicology is using newer technologies that are more sensitive, but you also have to be a little bit careful about this. We published a paper on metabolomics that showed that you could use it to look for biomarkers of estrogen response, in particular carnitine derivatives. But we also showed that a lot of the common statistical approaches to metabolomic data were incredibly misleading. And this is extremely important because, you know, bad data in toxicology leads to bad hazard assessments, which leads to problems later on. Another thing that we did was use 21st century toxicology to focus on human safety. So we use TCGA data, which is a very, very large data set of human tissue. And we use this to demonstrate that a lot of primate-specific genes were consistently overlooked in the literature. And this was really motivated by the fact that, you know, we would very often get our grants returns saying, this is great, this is a cool idea, but you need an animal model. So we really wanted to kind of put some numbers on just how many genes you would be missing if you were using an animal model. And this is for cancer, which is a relatively well-studied disease. So the problem would only be more acute for diseases like autism. We showed that several genes that were important for cancer progression were simply not studied, and that a lot of gene annotations were just systematically missing a lot of biology. Or to put this in layman's terms, we think we have sort of the Google map for the human genome. That's how a lot of people sort of sell our new technologies, but the reality is we're basically operating with sort of a medieval map that has vast territories that don't really have any information. And this is important for toxicology because we can't really navigate benign design if we don't have a good map of the human toxome. So another topic that we're looking at is the history of regrettable substitutions. DDT is one of the prime examples. There's also bisphenol A, which has been replaced with BPS because we didn't really understand the molecular mechanism of toxicology. So we've been doing a number of publications looking at the history of regrettable substitutions, how toxicology can avoid these in the future. And we also had a paper on better metrics for sustainability by design that was done with me and Hans Plug, who's also been enormously helpful in keeping green toxicology and high quality toxicology in general at the Green Chemistry Conference. And last but not least, the future of green toxicology. We really want to help use a high-dimensional map, high-dimensional data to sort of map the human toxome better than we have in the future. We're going to use respiratory sensitization as a case study for using non-animal models, and also using 3D models and chemoinformatics to sort of improve our model accuracy. And I wouldn't be a good author if I didn't just briefly give a shout out to my book. So Green Toxicology has gotten the imprimatur of the Royal Society of Chemistry. The book should be coming out sometime in 2022, probably not at a bookstore near you, but on Amazon. And that's it.
Thank you very much, Alex, for that excellent presentation. I loved your map, your medieval map, Here Be Dragons. A nice analogy. And you've put us back on time, even slightly ahead of time as I see it, 11:15 or so here in Baltimore. So we'll move from green toxicology to what's labeled here as Cosmetic and TestSmart Program. And we will hear from Julia Fentem. We were originally hoping to hear from Tony Gaspari as well. I don't think Tony is with us. Are you here, Tony? I don't think so. So the floor will be entirely with Julia Fentem. Julia is the Director of Safety Science at Unilever. I'm probably blowing some of this. She worked for many years at Frame, which is where I met her. And she worked closely with Michael Bowles on pioneering validation and many other approaches. And she's one of the giants in the alternatives field. Julia?
Thank you very much, Marty. Thank you for the invitation to join you all today and to celebrate 40 years of CAAT. It's good really that Helena covered a little bit of the TestSmart high production volume chemicals piece before going on to the developmental neurotox focus, because that was way beyond my abilities. So I will focus on the cosmetics piece. And in 40 years, I think we heard from Alan and from Andrew, it was really the basis of the establishment of CAAT with that grant from the Cosmetics Trade Association in the US. But I would say, CAAT has played a leadership role on various important 3Rs topics, many of which are far, far broader and potentially more impactful than the use of animals in ensuring the safety of cosmetics. Having said that, it is with some dismay and frustration that we're back talking about cosmetics after 40 years, and it's a hot topic again after a relatively quiet period of time, at least politically and from a campaigning point of view over the past decade. Really, I want to focus a little bit on where we are now rather than going backwards, but I have put the highlights as I saw them from CAAT's involvement in the early years, right the way through to 2011. But last month, I think this area was sort of blown apart. And I think there's probably at least half of the authors of the paper in all text on this call. So I think we've probably got Jean and Constanza as well as Thomas. Where actually what has been left a little bit hidden and not talked about as explicitly as it is being now, the so-called reach loophole has been exposed. And that paper is a very detailed analysis which shows that there's at least 63 dossiers for which animal data have been generated since the cosmetics testing ban deadline, where we've got cosmetics only ingredients. So that's a little bit of the context for what's been happening over the past few months. So in a rather unique collaboration now, and I think Andrew alluded to it, the European animal protection NGOs have come together with brands and companies essentially to try to save the European Union and the UK cosmetics testing bans. And the background here is really a case that was brought by Simrise to the European Court of Justice. And it concerns two sunscreen materials which are only used in cosmetics, for which the European Chemicals Agency, ECHA, is mandating new animal testing. And that turns out to be really the tip of an iceberg of probably about 150 or so ingredients that have been used safely for many years in cosmetics and other consumer products, where ECHA is now requesting new animal tests. And given the fact that the EU citizens, NGOs, cruelty-free brands were all instrumental in campaigning for the animal testing bans to be put in place in the first place, they have joined forces again, and this time with broader support from the cosmetics industry. And there's been a launch of what's called a European Citizens Initiative, which is a mechanism by which if you get 1 million valid signatures from citizens across the key EU member states, then action needs to be taken by the Commission. What this really just highlights is that lack of alignment in the EU of the cosmetics regulation with the chemicals regulation REACH. But from my perspective as more of a scientist, what it shows really is the ever increasing gap that we're getting between regulatory testing requirements in the EU and modern safety science. So science that's based on novel approach methodologies, new exposure science, computational modeling, and informatics approaches. So whilst the US has invested behind what I would consider to be very groundbreaking, which was the toxicity testing in the 21st century strategy that was published in 2007, and which probably has quite a lot of the authors and those involved on this call. And then the US EPA obviously having a very clear NAMS roadmap and investing in building the technical capability and having targets in place in terms of reducing animal testing of chemicals. In the EU, we don't really have anything comparable to that. And where there have been scientific advances, they're not being implemented by ECHA. And the crux of the matter there is we have legislation that requires us to use animal testing as a last resort. We have a new scientific toolbox based around NAMS, and then we feel that we're actually being blocked from applying that where it could add value. And this has now become a business issue. So beyond the realms of just the science, and to such an extent actually that last week I was asked to give a seminar on what was happening to Unilever's investors and analysts. And that's probably a bit of a first that shows you where the impacts and the edge of this issue now lie. So I wanted to show this chart with me in Baltimore because it was the last time actually that I got the opportunity to travel outside of the UK, given the COVID restrictions. And it was a memorable short visit to Baltimore way back in November 2019 for the CAAT Symposium, which was celebrating 60 years of the 3 Rs. And at that time, we were discussing how we could build on the great work that was being done in the US by the US EPA and other collaborators. But, you know, just turn the clock forward and we're, what, 2 years down the track. And we're now talking about something very different, which is essentially how can we use that great analysis that's been published in Alltex? How can we bring together where we have the new science, the new toolbox, and hopefully use that as a stimulus for starting to bring together all interested stakeholders to actually work in partnership to address this so-called reach loophole and to use essentially and leverage what the work that the US EPA and others have been doing to hopefully try to influence and help shape some change in the EU chemicals legislation. If anyone can actually explain to me how animal test data that are generated under REACH actually connect to how we do health and safety control measures in our factories, which actually be based on detailed local exposure-based risk assessments, then I'd very much like to know. I think that's the crux of the matter, that we have a hypothesis that shows data that's generated under the REACH legislation and somehow think that that is protecting workers in factories. Well, in Unilever, where we use an end-to-end approach, and obviously I have that safety responsibility, we don't feel that we need new animal data as part and parcel of how we best protect the workers in our factories. So I think I'd just like to share, I think the latest news coming out of CAAT from the European side of things, and that is that the Alltex publication is going to be followed up by essentially shaping a new chemical safety without animal testing policy initiative and planning an action platform for cosmetic products without animal testing. And I think this is really what consumers and retailers and brands and companies want. But it's going to mean tackling some current traditional thinking and current policy in terms of how we best assess the safety of chemicals and their use in consumer products. So we have this completely new and ever evolving NAMS toolbox, transcriptomics approaches, cell stress panels, biokinetic modeling, pharmacological profiling, What we need is progressive chemicals regulations in the EU, which facilitate their use such that we can potentially do weight of evidence and risk-based decision-making. And finally, I'd just like to congratulate the CAAT team on a very successful and productive 40 years. Lots of highlights. Many of those were on display at the recent 11th World Congress. And I think CAAT is founded on open collaboration and on taking action. And since 2010, obviously has strengthened its transatlantic partnerships and influence. And this is an excellent foundation for the years to come. I think events of the past few months have shown that this is definitely going to be needed in the years ahead as we seek to develop innovative and more environmentally sustainable new materials. We just had the nice session on green toxicology, but we need to ensure that the safety is using equally advanced and innovative non-animal safety science. Thank you.
Thank you, Julia, for that excellent presentation. And I think it harkened back to Andrew's comments about the changes over the last 40 years where the animal groups and the companies were at each other's throats. And here we are 40 years later with them working together to try and stave off a retrograde development. So things have changed, and hats off to CAAT for stimulating a lot of that change. So we're at the point of a break. We're nicely back on time, in fact, with a couple of minutes to spare. So let's reconvene at 11:40 Baltimore time. And you can maybe make sure you're muted while you're taking a break. So see you all in 10 minutes or so. Okay, we'll start in a minute. Please start to settle in. Okay, I have 11:40 Baltimore time at this end. So let's resume. Again, I'm Marty Stevens with CAAT, your moderator today. And I would just take a second to encourage those of you who are on the call to use the chat function, especially if you go back many years with CAAT and would like to note an anecdote or a short story of your involvement with CAAT. Okay, now is the time for the session on the education program. And we'll be hearing from Deborah Rudacille and Lena Smirnova. Deborah worked at CAAT. Deborah Rudacille worked at CAAT during its earlier years. She wore many hats then, but I think her primary role was as a science writer, in which she authored or co-authored many publications, including monographs. Lena is a scientist with CAAT, active in the lab, using the mini brain model. She directs CAAT's education efforts, including some very popular courses online at Coursera that anyone can avail themselves of. So Lena and Deborah, the floor is yours.
Hi, everyone. Can you hear me?
Yes.
Okay, great. Thanks so much to the organizers for inviting me to be part of the celebration of the 40th anniversary of CAAT. It's really great to see so many old friends today, as well as to get caught up on some of the new developments since I've been away for quite a while. My tenure at CAAT began in 1992, roughly a decade after its founding, when Alan Goldberg and Joanne Zerlo hired me to serve as the scientific writer in CAAT's education program. I came to CAAT as a very early career science writer, having previously worked as an editorial assistant in the Department of Neurosurgery at the University of Maryland. At the time that I was hired, I probably knew as much about the uses of animals in research and testing as the average American. And I was probably a good candidate for the job because I really didn't have strong feelings on either side at the time. I will always be tremendously grateful to Alan and Joanne for hiring me as a fairly green science writer for the job, as the 5 years that I spent at CAAT really launched my career as a science journalist, a scholar, and more recently as a college professor. I really doubt that I would have been awarded a Guggenheim Fellowship in science writing in 2017 had not Alan and Joanne taken a chance on me as a newbie science writer back in 1992. So when I started working at CAAT, our offices were on the first floor in the Johns Hopkins School of Hygiene and Public Health, building on Wolf Street. It was a great place to work, and its history was really a major part of its allure for me. Early on, I became very interested, not just in the history of the school, but also in the history of medicine and public health. And I spent a lot of time across the street in the Welch Medical Library, particularly after Alan, Joanne, and I began working on the manuscript that became Animals and Alternatives in Testing, History, Science, and Ethics, which was published by Marianne Liebert in 1994. I learned so much from working with Alan and Joanne on that project and via my independent research. And I later realized that my experience was kind of similar to being a graduate student in the history of science, learning about the history of animal experimentation, the early development and use of cell, tissue, and organ cultures in the 19th and early 20th century, and the role that both in vivo and in vitro methodologies played in vaccine development and more broadly in the development of scientific medicine. I also learned a great deal about the history of toxicology and how and why various tools and techniques came to be used to establish the safety of a broad range of consumer products and drugs, and why it was essential to replace them as science developed powerful new tools and techniques. And of course, I learned about the principles of humane experimental technique and the two pioneers of the movement for humane science, WMS Russell and Rex Birch, and their successors, who I met over the course of my involvement with CAAT. In addition to working on the book, Animals and Alternatives and Ethics, I also served as editor of the quarterly CAAT newsletter, which in those pre-internet days was our primary outreach tool. And I recall that it had a circulation of more than 40,000. Alan, you can correct me if I'm misremembering that, but that figure sticks in my head. I worked closely with the newsletter editorial board, soliciting articles from experts in the then controversial field of alternatives. And the most important lesson that I learned in those early years at CAAT was that it was possible to both support animal research and also to work for the replacement, reduction, and refinement of animal use in science, and that implementing the 3 Rs in research protocols would produce more accurate data. Today, that seems self-evident to all of us. But back even in the 90s, beyond those really crazy years in the early 80s, the debate around animal use in research and testing was highly polarized. And CAAT's perspective was a truly revolutionary perspective. In fact, as I learned when I read Thomas Kuhn's The Structure of Scientific Revolutions, It was a paradigm shift of the highest order. But even before I read Kuhn, when I was working on the Lieber book, I was learning that animal protection and support for research were not opposing principles, but complementary ones from the people I met at CAAT workshops, advisory board meetings, and at the World Congress on Animals on Alternatives in Testing. So here's an anecdote. At one of my first CAAT workshops, I believe in 1992, I noticed this kind of rumpled, white-haired man in shirt sleeves, baggy trousers, and tennis shoes who seemed to be in constant motion, roaming around the hall, while everyone else pretty much stayed put in their seats. So I was really curious to know the identity of this seeming fish out of water and went up and introduced myself. And Henry Spira then invited me to sit with him at lunch. Henry, as you all know, played an enormously important role in the founding of CAAT and later the Center for a Livable Future. And he became a really important mentor to me. From Henry, who had a very long history in various social justice movements, I learned that outsiders and insiders need to work together to create deep and lasting change, that activists force institutions to consider changes, which are then implemented by those working inside institutions. The history of CAAT and its enormous success in changing the culture of science, I think, bears out Henry's thesis, one that I've seen play out in many other social movements since then. I'll share with you a quote that Henry wrote in 1993. Many people perceive the movement for animal protection as negative and anti-science. To counteract this perception, we have continually accentuated the fact that members of the scientific community are part of the 9 out of 10 people concerned with protecting animals. We have attempted to turn walls into bridges by promoting the shared goals of better science, efficiency, economy, and humanity. And I have to say, if I retain any hope that we will overcome the challenges that we face today, it's by reminding myself that Henry Spira and Alan Goldberg built a bridge across the abyss that separated scientists and animal rights crusaders in the early 1980s. One that seemed just as unbridgeable back then as the one separating scientists and supporters of science from anti-vaxxers today. During my CAAT years, I was also really thrilled to meet W.M.S. Russell, the Oxford-trained zoologist who co-authored the Principles of Humane Experimental Technique, and who seemed to me to have stepped out of the pages of a 19th century English novel with his hand-knitted jumper and tweed jacket, speaking the then nearly forgotten language of scientific humanism and bridging two cultures, which were sadly estranged by the time Russell and Birch wrote their astonishing little book in the 1950s. Although I only met him the one time, Bill Russell's example of bridging the two cultures of the sciences and the humanities deeply influenced me and remains a model that I strive to emulate in my own research and teaching today. Others associated with CAAT who I remember with great fondness and whom I learned important lessons, apart from Alan and Joanne, of course, include Andrew Rowan and Marty Stevens on the animal protection side, Steve Gettings and Kevin Renskers on the industry side, and veterinary scientist Neil Wilcox and Bill Stokes of the Interagency Coordinating Committee on the Validation of Alternative Methods on the regulatory side. All of these people had different perspectives and different professional goals and responsibilities, and yet they all came together for a common purpose: to develop a more precise, a more efficient, and a more humane science of safety testing. And this was great training for me as a science writer, because it gave me experience in seeking out and balancing sources with different points of view and different fields of expertise to create a balanced story. And I often tell my science writing students today, ignorance can be an enormous asset for a science writer because we're starting in the same place as most of our audience. And we have to learn the science ourselves in order to explain the science and social context of the science to our readers. And we science writers are really fortunate, because who do we learn the science from? We learn it from the acknowledged leaders and experts in their disciplines. And what a privilege it was to learn from the alternatives leaders and experts I met during my years at CAAT. So I'll tell you a little bit about the last significant project I undertook at CAAT, the Catalyst newsletter for middle school students, which was a critical component of the education program spearheaded by Joanne Zerlo and founded by the Geraldine Dodge Foundation. We were aware that middle school students were very concerned about the use of animals in research and testing because they sent us letters. They would be doing projects at school and they were really craving information. And none of our existing materials at the time, not the CAAT newsletter, not the Liebert book, not the scientific papers produced by CAAT grantees, was suitable for these young readers. So Joanne and I conceived this idea of producing a comic book. My son was really into manga at the time, so that might have been partly the trigger for this. So our comic book had a heroine named Dr. In Vitro, who would explain how and why animals were used in research and testing and which alternatives could be used to replace, reduce, and refine animal use. Joanne and I worked with two wonderful graphic artists to produce that comic book, and I believe we produced maybe 4 issues. We not only mailed that comic book out, but we also took our show on the road. For about 3 years, Joanne and I traveled all over the country to scientific meetings, as well as annual meetings of science and biology teachers. We staffed the CAAT booth and exhibition halls around the country. We hung up our CAAT banner, set up tables with our educational materials, and answered questions about alternatives. And I'd like to think that we made a few converts at those meetings, as well as connecting with people who already supported CAAT's mission. We certainly had a lot of fun on those trips. And on one of our final trips to Pittsburgh, I had an important outcome for me personally. While we were in Pittsburgh, I had a dream, which I couldn't remember when I woke up, but the phrase, "the scalpel and the butterfly," was in my mind, and I scrawled it on a piece of hotel stationery and put it in my briefcase. Within a year, I had pitched a book with that title about the long history of the conflict between animal protectionists and experimental scientists to a New York agent who sold the book to a New York publisher. And that book, "The Scalpel and the Butterfly: The War Between Animal Research and Animal Protection," which was published in 2000, is in some ways my doctoral dissertation built on the research, skills, knowledge, and contacts I acquired at CAAT. I would never have written that book if not for the 5 years I spent there. And all of my subsequent books, as well as my 25-year career as a science journalist, rests on that foundation. So I'll end this talk by once again thanking Alan Goldberg, Joanne Zerlo, and everyone else associated with CAAT for all you've given me personally, as well as all you've given to science and to the world over the past 40 years through your cutting edge science and promotion of ethical animal care and use. If science and animal protection are no longer at war, it's in large part due to CAAT and those who contributed to its mission. I feel really honored and so grateful to have played some small part in that history. And I thank you all. Bye-bye.
Thank you, Deborah. Lena?
Yeah, I'm taking from here. Thank you. This was a very great journey through the 90s, 2000 of CAAT. I will update you on what CAAT is doing currently in terms of education program. And of course, first of all, what our goals are is to disseminate the information, collect the knowledge, and disseminate the knowledge. And of course, the targets we are reaching, it's far beyond academia. We are talking also regulators, industry, and also not to forget general public, or for example, schools, how Deborah mentioned already. And what we have been doing over the last couple of years, or 10 years, 12 years, trying to respond to the increased demand in education and how people want to learn about alternatives more and new approach methodologies, expand the variety we can offer, how we can teach this field, and of course further increase outreach. And I will go through
starting from academia, going quickly through the classes which are offering, CAAT faculty is offering mainly for master and PhD programs in Johns Hopkins University, and also how we go further beyond that. So this list of classes which Katrin Herman is teaching, and she is our director of the refinement program at CAAT, and she will be talking in more details about those classes. I just wanted to say that Katrin did a great job on revamping the existing courses on animal research, law, policy, and humane sciences. She introduced the refinement of animal experimentation course and animal ethics. And she is loved by students, which is also supported by the excellence teaching she has been receiving over the years. She has been teaching those classes, so they're very popular, and Katrin will talk more about them. She's also co-teaching a certificate on humane science and toxicology together with Dr. Paul Locke, who also will be speaking today, and he is associate professor at our school and also policy program director. You already heard about green toxicology from Alex, but Alex also is a great teacher, teaching our new scientists how to use proper statistics, how to use bioinformatics, and how to deal with big data we are surrounded in the classes she is teaching, and also receiving excellence in teaching from the students she is supervising. Myself, together with Thomas Hartung, we established 2016 2 courses on toxicology of 20th century scientific application and evidence-based toxicology, which is the series of lectures which are online, and we teach them in master's and PhD programs. And I must say that a lot of CAAT faculty are also involved in those courses, and we received several years in a row excellence in teaching from our students. But most importantly, we use the opportunity which Hopkins also allows us to bring those courses to broader audience beyond Johns Hopkins, so you don't need to pay a crazy tuition and just enjoy those courses for free. Both courses are on Coursera. This is a massive open online course platform. It's absolutely for free. And for example, if you go on Coursera and put in the search TOX for talks, the first class you will pop up, it will be toxicology of 20th century. And we have currently over 13,000 active learners in these 2 classes. So we consider it as a big success and a great outreach to the broader audience. We also teach not only in Hopkins, but also outside the Hopkins by giving different lectures at different universities. And one of the as a block of class we are teaching right now is in Georgetown University as a master program on environmental metrology and policy, which is co-organized with US EPA. And they requested introduction to alternative to animal testing for those students. And we are happy to teach this class also outside the Hopkins. Teaching alternatives outside of GHU is, of course, also it's all the workshops, webinars, summer schools, and so on, which CAAT is organizing. And one of the examples I put on this slide is summer school in the middle of pandemic in 2020, where Katrin Herman was heavily involved from our side, and CAAT co-organizes with PCRM, where we reached over 600 attendees. I consider that as a huge success, and it's supposed to be in person. It was online due to pandemic, but that way we could reach people at different continents, different countries, and also students or scientists who would be not able to travel to US to attend this summer school. And of course, teaching through research. This is the big part. Also, I'm involved by supervising students in the lab and their research projects from master to PhD and postdocs. And of course, teaching through research is not only singing and puppeting in the lab, but also a lot of fun. We have a lot of parties, and we are a big CAAT family with our students. And we have, through our outreach, we get a lot of movements from different countries, people reaching out to you and try to would like to come to do the master thesis or internship from different countries. And I listed few of them, and I put only some pictures because I don't have space on the slide to put all the students who visited our lab. I would like to close that, just to repeat again that increased demand on learning about the alternative, and our dissemination strategies and outreach strategies. This is help us to educate new generation of scientists, to increase the outreach and to move the field of 3Rs forward and the paradigm forward. Thank you very much.
Thank you, Lena and Deborah, for those excellent presentations. Clearly, educating the next generation of scientists is of critical importance, as well as educating other stakeholders on these issues. So we're a bit behind time now. If the remaining speakers can help us gain back any of that time, that would be appreciated. Our next topic is refinement, and we will hear from Joanne Zerlo and Catherine Herman, names that you've heard numerous times already. Joanne was a scientist at CAAT for many years, spanning the periods when Alan Goldberg and Thomas Hartung were the directors. And among other things, she directed our refinement program. And Catherine Herman is a scientist at CAAT, directing our refinement efforts, as well as championing human relevant non-animal methods. Dr. Herman recently began a parallel second job as Animal Protection Commissioner in Berlin. Joanne, looks like you're queued up next.
Okay, I hope everybody can hear me. Anniversaries are a great time for both looking back and assessing progress and looking forward to planning the future. I guess 1981 was a good year to start endeavors. I just wanted to say that in addition to CAAT celebrating its 40th anniversary, Jim and I marked our 40th wedding anniversary in July. So I guess that's a good omen. In my case, as Marty just mentioned, I'd been a part of CAAT 1.0 under Alan, and then after a 10-year stint at the National Academies, I returned to CAAT 2.0. I hope you'll indulge me a little bit with a little perspective and my own personal growth as a scientist and as a person, in putting this talk together, reminiscing about some of the things that I had experienced. I started with CAAT in 1990 as the assistant to the director, who of course was Alan Goldberg. I came from a purely laboratory-based background, having participated in cancer research that was typically at the in vivo, in vitro interface. And I worked with primary cells or cell cultures to study mechanisms of chemical carcinogenesis and toxicology. I really hadn't anticipated the direction in which my career would go or how many aha moments I would experience along the way. Probably the first aha moment was the 3Rs, totally new to me. I had never heard of Russell and Birch, didn't know that there was an active group, albeit small, working to find in vitro alternatives for toxicity testing. In fact, even though I had come from a department of pharmacology and toxicology, I'd been focused on the mechanistic research processes and really had little knowledge of applied toxicity testing at all. However, I started at CAAT by coordinating the grants program, so I did know a little bit about writing grants. And I eased my way into the field. I had great mentors along the way, including John Fraser, whose picture you saw before, uncredited with me in the laboratory, Alan, of course, and members of the CAAT board, many of whom I became very close to over the years. Probably one of the most memorable experiences I had was participating in the 1995 Sheringham meeting on the 3Rs, where I had the honor of meeting Bill Russell and Rex Birch. And I know many of you who are participating today were at that meeting also. It was a game changer for me and really opened my eyes. So Deb already talked a lot about the education program. Alan and I was asked to elaborate on that a little bit too, in addition to the refinement program. But Alan and I had the idea to build the CAAT education program, and we hired Deb, and it was a great collaboration, as you just heard, producing the Catalyst newsletters and the comic book. It was through these activities that I had another great aha moment in recognizing what I now see as obvious, that A, the public has very little knowledge and appreciation for science, and B, scientists have in general a poor track record in communicating their work with the public. Using the 3 R's as a basis, we tried to communicate some basic science information about science in our publications to students. And in reaching out to science teachers, we hope to give them some material that would enhance their ability to convey how science is really done and how discoveries are actually made. These activities led to my participation in several ongoing efforts by AAAS and SOT to improve the public communication of science. So another aha moment that I had was recognizing the reticence of scientists to even consider the 3 R's as anything but a threat to their scientific freedom. Compared to working with students, talking to scientists about the 3 R's had the effect of marking me as one of them, someone sympathetic to the animal rights movement, and decreased my credibility quite a bit, even though I was working with an academic center. That really hit home to me after I was hired from CAAT to direct the Institute for Laboratory Animal Research, or ILAR, at the National Academies. One could hear at meetings the undercurrent of conversations when I spoke publicly, like I was out to subvert the entire research enterprise. Here I was back on the learning curve again. Through my work at ILAR, I quickly learned that there was a very caring veterinary community serving the research enterprise that cared deeply about the animals being used in research. In other words, these professionals were deeply involved in one of the 3 R's, refinement. Again, something about which I knew relatively little aside from my conversations with people like David Morton and Bert Van Zutphen, whom I had met at Sheringham. Another aha moment for me was that while we could work to replace animals in research and testing, the goal would not be accomplished in the near future. The animal research enterprise was still using millions of animals per year, and considering the welfare of those animals was critical, both for the animals themselves and for the integrity of the science. When I was offered the opportunity to return to CAAT in 2010 under the new leadership of Thomas Hartung, one of the areas I wanted to develop further as part of CAAT's mission was refinement. There were some basic refinement activities going on, including the grants program, about which I'll talk about in a minute. I felt that CAAT, which had always maintained its scientific roots, could contribute greatly to the incorporation of more humane methods in both research and testing. This effort required the collaborative effort of the veterinary community, and in the process of setting up the program, I capitalized on my relationships with veterinarians from government, industry, and academia from my years at ILAR to highlight easy ways to bring improvements to the conditions of laboratory animals. In that time, the field of lab animal behavior had been growing, and more scientists were dedicating their careers to understand the behavioral needs of animals in the laboratory setting, including caging requirements, environmental enrichment, and social housing of social species. New findings were showing that scientific results of animal studies were clearly affected by the emotional and physical state of the animals. So there was really a scientific incentive for improving the conditions of learning about the needs of laboratory animals and improving their conditions. Veterinarians were typically aware of many of these improvements and their scientific implications, but typically the scientists that were harder to convince, not really believing that one could assess the quote unquote happiness of an animal, even though the methods for quantitatively assessing animal welfare were advancing considerably. Thus, the refinement program focused on holding workshops and interactive meetings to bring together those doing research on animal behavior with those using and caring for animals in the research setting. We established a continuing series of workshops on social housing of animals, with each meeting addressing the needs of various species, both with presentations from experts and interactive discussion sessions. The refinement program also built upon the small grants program begun by Alan, as I mentioned a few minutes ago, to award small amounts of money to those working to improve lab animal welfare. We awarded 3 to 4 grants a year to those working in diverse species ranging from fish to non-human primates. While the amounts were small, some of the investigators were able to conduct very focused studies that could have a significant impact on animal welfare. The refinement program has evolved in the almost 4 years since I've retired, and I will now turn the presentation over to Catherine Herman, who took over for me and has moved the program forward. I thank you all for your attention and for the honor of being able to share my thoughts on this special occasion. Thank you.
Thank you, Joanne. Catherine?
Catherine, you might be muted.
Still can't hear you.
Now?
Yes.
Okay, I'm sorry about this. I have to share again because I should have unmuted before. Sorry, I couldn't unmute myself anymore. Sorry, let's try this again. I should be more familiar with this by now, sorry. Okay, now, thank you, Joanne. Yeah, actually, I'm pretty new still. I feel new still at CAAT because I only followed in the footsteps of Joanne at the end of 2017. And first of all, I want to give a little bit of an insight about my background because that also led to me changing the program a little bit and calling it now the Beyond Classical Refinement Program. So before I came to CAAT, I was a federal regulator in Germany. And what I did there was that I assessed animal research proposals for the government, and I also inspected countless breeding facilities and animal laboratories. And I came into this position quite naive as somebody who wanted to help individual animals that were used in the name of science. But because I did this job for several years, I became more and more aware of the considerable harms to the animals, because I also realized that the refinement methods were often not applied in practice. So the animals did not benefit from our refinement research. And I also saw that many of the animal research proposals actually lacked scientific rigor. Something we talked a lot about today is that we need high quality science. And so that actually made me extend the refinement program as it was before, the classical refinement program, because I thought it was very important to include the refinement of planning, conducting analysis and reporting of animal experimentation. And that is also something that then I focused on in the teaching. So also in the refinement course that you will later see, one big focus is quality of research and how to achieve that. Because as we all know, we are in a so-called reproducibility crisis, and about 50 to 90% of the preclinical animal studies are not even reproducible. So we don't even talk about translation, just reproducing the same study. So to counteract the reproducibility crisis, I included this other refinement of, to improve the science in the program. And I wrote this extensive paper. If you want to know more about my thoughts on refinement, the current state of refinement, it was published in 2019. Another thing that I think is an important refinement is the culture of care. And that's also something we promote among our students and early career scientists. Lena already talked about the 3 courses. I just wanted to highlight, for example, the Animals in Research course. That's one course we revamped, and many of CAAT's scientists are actually giving lectures there and in the other courses as well. So it's a collaborative effort. And the idea here is to build bridges to show the students that there are not only animals available to do research, but also the non-animal approaches. And we discuss what are the shortcomings of all the methods. And another focus we have in this Animals in Research course is that we have an extensive focus on literature search, especially for NAMs. that's a big part of the course. So the students can find the best method for to answer their research question, and that's often not an animal model. And I just wanted to briefly talk about our award. We used to have a refinement award, and that was also extended a little bit to reduction and refinement. And that had to do with the possibility, to further reduce animal experiments by, for example, doing systematic reviews before or instead of doing more animal experiments. So we gave grants for that, or we are giving grants for that. And also we funded a preclinical research platform where you can pre-register your research. That's also something we promote, because obviously that can reduce animal numbers and by not duplicating studies that were already done. And this year we actually awarded the research for an in vitro and in vivo extrapolation, and that was something we never did before, but that's also in the reduction R. I don't know why this doesn't. And also, Lena already pointed out that we do much more than just university teaching. And one other thing I started this year is a monthly 3 hours training series that's actually free of charge, and it's addressed to scientists, but also to IACUC members, or in Europe, you talk about animal experimentation committee members, and the so-called competent authorities in Europe who are authorizing or assessing and authorizing animal research proposals. And here the idea is also to really bring together people, because that's one thing we are facing. We have to bring together people working with animals and people who work with non-animal approaches to have a better dialogue and bring people in the same room. And this webinar series has been quite successful. It has had a lot of interest. If you are interested in that, it's always advertised in our newsletter, and also you can find it via Eventbrite. Yeah, so that's actually what I wanted to share. Oh, sorry. And happy birthday to CAAT. I'm very happy to be part of this center. Thank you.
Thank you so much, Catherine and Joanne. We're a bit behind time, so we're going to go right into our next talk. We're very privileged to have Bert van Zutphen with us from the Netherlands, who will be talking about the World Congress on Alternatives. Bert directed the Animal Care Program at the University of Utrecht for many years. Prior to his retirement or semi-retirement. He co-organized the very first World Congress on Alternatives held in Baltimore with Alan Goldberg, and he was the primary director of the second World Congress, which was held in Utrecht. Bert?
Bert, you're muted. You're still muted. You're sharing, so your commands should be on the top of your screen, perhaps, if you just hover over to the top of the screen.
Bert, you're muted.
I hope
there
you can.
Go. Yes.
Yeah, okay. Can you hear me now?
Yes.
Okay, great. Well, thank you for inviting me. Thomas asked me whether I could say some words on the World Congress, and in particularly the role of CAAT in initiating the World Congress series. And I think that's a very appropriate thing to take in this meeting, in this meeting. Well, when I start talking about the World Congresses and in particularly the role of CAAT, I think I have first to indicate the role of Alan Goldberg. Alan is not only the founder of CAAT, but I would call him also the father of the World Congresses on Animal Alternatives. Alan, I would like to talk a little bit on how it started. The format of the first World Congress as basis for subsequent congresses. I also would like to say some words on the 3 R's, because they have always been very important for the framework of all the congresses. A little bit on the rewards that are mentioned during the congresses and presented to many of the persons who are here now in this meeting. And at last, I would also say some words on the Alternative Congress Trust. Well, let me first say how, from my perspective, how it all started. Alan came to Utrecht in 1990. He visited me at my department by then, and we had lunch together. And during this lunch, we discovered that our ideas on the ethics of the use of animals in research and testing were very similar. And well, at a certain moment, Alan said, "Bert, I have a dream. My dream is to organize a World Congress on Animal Alternatives." And I said, "Wow, how do you think to do this?" He said, "Well, it is now 1990. In 3 years, we could have a program together, and then we can organize a World Congress on Animal Alternatives in Baltimore," which was a nice idea. But then he said, and what I would like to see is that you would be my co-chair for that Congress, and then organize the second World Congress in Utrecht. And then I hesitated a little bit, but I found it a very well idea. And since it was still 6 years from 1990, so I said, okay, I take the challenge. And so it started from my perspective. Alan went to Baltimore. I think it was at the 7th symposium, CAAT symposium, that he presented the idea to an international group of experts. And the program was put together. And I will make it short because the program is still similar as the programs as they are now in all the World Congresses. The themes, the topics were by then in vitro studies, risk assessment and extrapolation, validation, databases, education and training, animal welfare issues, ethical aspects, and barriers to the acceptance of alternatives. And most of these, as I said, themes are still represented in successive World Congresses. Alan had also a lot of sponsors on board, like Procter & Gamble, Unilever, L'Oreal, the Humane Society of the US, and 20 more, I think. And also, many of these sponsors are still supporting the World Congresses. There is a nice publication on the World Congresses from, I cannot see it myself here because, but it's the publication indicates that the first World Congress was organized in Baltimore by Alan Goldberg and myself as co-chair. And then the second World Congress, which was organized in Utrecht, and I was lucky that Michael Bols would be my co-chair then, and Michael, in turn, organized the third World Congress with Andrew Rowan as co-chair. Andrew, in turn, choose for Horst Spielmann. And so it went on and on until the 10th or 11th World Congress that we have had this year. So there was a very smart idea to have a co-chair who would then be the next chair for the next Congress. The proceedings of the World Congresses were firstly published in a book. Marianne Liebert published the World Congress on Alternatives and Animal Use in the Life Sciences of the Congress in Baltimore, and Elsevier published the next two congresses. And there were huge books with more than 1,000 pages. But the proceedings of the next World Congresses have been published in ATLA and afterwards in Altex. Where Alan is the father of the World Congresses, we could say that Bill Russell is the father of the 3 R's. But when he published his book in 1959, I think it was not so well known in the beginning. That changed in the last 80s, early 90s, and in particular when he was vice president of the Humane Society, when he established the Research on Bird Award, that was a good point to make it more well known. And in 1993, when we had the first World Congress, was Bill Russell by Alan invited as a guest of honor. And that was the same was true in 1996 and 2002, when we had congresses in Utrecht, Bologna, and New Orleans, respectively. So there was Alan, mostly plenary speaker, but not only a speaker, he was also a singer, but mostly he presented a song during his presentations, and even once in a while he was dancing like in Bologna. So here we see Bill, I said Alan, I meant Bill, of course. Bill, we see Bill here with his wife Claire, and if you look well, this is Alan and Helena in 1996, so Alan quite a time ago. I have one picture more of Bill. We visited Bill Russell, Anna-Marie and I, my wife Anna-Marie and I visited Bill in June 2006. We went for dinner or a late lunch, I think it was. Anyway, we had nice talking, and Bill talked about his life, but also about his health problem. He had kidney problems, and he had to go to the hospital every 3 days for dialysis. And you can imagine that we were shocked when we get 6 weeks later the message that Bill died. And this is probably the very last picture of Bill Carey taken during his life. So I would like to show you that picture during this meeting. His name is, of course, still very well presented in the World Congresses, because Alan got the first, that was not during World Congress in 1991. But then Konrad Hendrikse got the Russell and Birch Award in Baltimore in 1993, Andrew in Utrecht, and Katrien Stitzel in Bologna, David Morton in New Orleans. I got it in Berlin, Roger Curran in Kyoto, Thomas in Rome, Julia Fentem in Montreal, Marty in Prague, and well, the last one was Marx in Maastricht. But there were many more awards during the World Congress, like you see here in the Second World Congress in Utrecht, the Doornkamps-Binden Prize was presented to Albrecht Wendel and Thomas Hartung. I think it was for their work on pyrogenicity testings. Catherine just mentioned the CAAT Refinement Award. CAAT had also in 1996 the CAAT Recognition Award, which was presented to Michael Vestin here, by Alan, and in the presence of Prince Laurent. He was one of our guests at the Second World Congress in Utrecht. So, but there are many more awards, but I cannot go into the awards now because I would like to say still some words on the EC, the Animal Congress Trust. I think the Animal Congress Trust was first founded by Andrew Rowan in 2001, but can be 2002, I'm not quite sure. But one of the reasons was that when I organized the Second World Congress, I got $75,000 from Alan to start the Congress, because we needed that money, otherwise it would not have been possible. That was partly the leftover of the first Congress. And I, on my turn, handed the $75,000 to Michael Bolt so that he could start the next Congress in Bologna. But that was, of course, not the best way to organize congresses. So a more structural basis for the World Congresses was presented by Andrew by establishing the ECT. But the ECT has more goals, like educating scientists and the public regarding alternatives, to promote wider application of alternatives, organize meetings that will contribute to understanding, and also getting consensus regarding alternatives, publishing proceedings, and cooperate with other organizations. The ECT has a board and 5 ambassadors. The chair is Elaine Fausman, and treasurer is Thomas Hartung. ECT provides a financial basis for the continuity of the World Congresses. As I said, it is more than the 75,000, but this is, we talked about 30 years ago. So that was quite an amount, and it was very nice to have it. Congresses are now, I think, cost more than $1 million to organize it. Anyway, I would end this small talk by this slide showing the friendship, not only professional, but also on a personal basis between Alan and myself. And I would like to congratulate not only Alan, but also Thomas and all the former and present workers at CAAT for their tremendous good work that they have been done and are still doing. And I can say a lot have been done, but we know all that still a lot of work has still to be done. And I wish them all good luck, all the success by this work. Thank you.
Bert, thank you very much for those kind words and for those historical reflections. I'm glad we're recording this so we have this for posterity. So we're now up to the policy program on the agenda. And I would note that we're running a bit behind in the schedule. And again, if any of the speakers can help us make up, would be appreciated. The policy program segment will feature Paul Locke and Pamela Frosch. Paul is a professor and lawyer here at Johns Hopkins University. And for many years, he's directed CAAT's policy program, which he'll talk about today. And Dr. Frasch is the Brooks McCormick Jr. Scholar of Animal Law and Policy and founder of the Center for Animal Law Studies, CALS or CALS. CALS is an academic animal law program with a focus on academic research, scholarship, and experiential education. The mission of CALS is to educate the next generation of animal law attorneys and advance animal protection through the law. So I turn the floor over to Paul and Pamela.
Thank you. And I believe it is my privilege to be able to go first. And I'd like to congratulate the Center for Alternatives to Animal Testing on its 40th anniversary, and to share just a few thoughts about the impact CAAT's policy program has had on the work of animal advocates outside of Johns Hopkins and outside the scientific community. I was first introduced to Drs. Paul Locke, Thomas Hartung, and Alan Goldberg almost 15 years ago through a mutual supporter of CAAT and of my program, CALS, at Lewis and Clark Law School. And these supporters intuitively understood the connectedness of the work CAAT and CALS was doing and facilitated our meeting in the hopes that some collaborative and useful projects would bubble up. And indeed they did. So prior to this connection, lots of attorneys and scientists seemed to work in relative isolation on many of the same issues, without the benefit of understanding each other's language, the levers for change specific to their particular constituencies, or the power that can be harnessed when different disciplines come together to work toward a common goal. But because of the CAAT policy program, animal law attorneys at CALS and beyond, who are working on animal testing issues, are being educated on the science and provided meaningful support, expertise, and encouragement in their work in the legislative and policy arenas. As a result of this cross-pollination, I and many others in animal law have now had the opportunity to co-write and present legal and scientific papers with CAAT scientists, co-organize and present at numerous interdisciplinary symposia exploring animal research issues from a variety of legal, policy, business, and scientific perspectives.
Pam, let me interrupt for a second, sorry.
Yeah.
There's a technical issue in that many of us are still seeing Bert while you're giving your presentation. And as much as I admire looking at Bert, I think we should see you on this, and perhaps the people behind the curtain can help make that happen. I have no idea how to do that, but in the interest of time, I think you should continue, and hopefully we'll resolve that problem. And in the meantime, Bert can maybe lip sync your presentation. All right.
I think we are all enjoying looking at Bert, so I will continue on and I'll just be in the little box at the top, and that's fine with me.
Okay.
Right ahead.
So I did want to say that we've also been able to co-testify in front of congressional committees and co-create a number of unique resources to foster further collaboration and understanding, including, for example, a glossary of legal and scientific terms so that we can actually understand what the other is saying, and that's been hugely impactful. So the widespread impact of CAAT's policy program cannot be overstated. Those of us working in this space now have access to cutting-edge scientific information, and perhaps more importantly, the privilege of working with CAAT policy program scientists and staff who help us understand and interpret the science so that when we do go and work on policy and legislative proposals, we know that we're bringing the latest information from the most authoritative source. And of course, CAAT has had a direct impact on policymakers through its innovative lobbying program. All of these efforts have been game-changing for attorneys and other advocates. And as we continue these and new collaborations, I look forward to the next 40 years, although I probably won't be around, but I look forward to the next 40 years of legal advocates benefiting from the tremendous work of CAAT. So thank you, Kat, for your essential work and happy birthday. I'd like to turn it over to Dr. Locke.
Thank you, Professor Frosch. And I wanted to tell everyone it's an honor and a privilege for me to be here to talk about the policy program, which originated at CAAT. I also just want to note that I'm going to start my presentation at the beginning, which at least for me was about 21 years ago, around the year 2000, when I was new to Hopkins. And I came to Hopkins as part of a leadership team of something called the Pew Environmental Health Commission, which was established at the School of Public Health to bring greater attention to environmental health issues and how to address them on a national scale. Now, the commission was an 18-month project, and towards the end of that 18 months, I met Dr. Alan Goldberg, who was at that time leading CAAT. So my personal involvement with alternatives issues was initially sparked by a hallway conversation I had with Alan. And after learning about CAAT and its, at that time, 2-decade work on the 3 R's and alternatives, I was very excited. And for those of you that know Alan, you know that a hallway conversation with Alan is a great treat. And he has a way of getting you all excited about alternatives, the 3 R's, and the work that CAAT is doing. One of the reasons I was excited, though, is because as an environmental health professional, I realized that replacement alternatives in particular, those that didn't use animals, could address many of the shortcomings in our current approaches to chemicals regulation and drug development. 2 things immediately came into focus for me. The first is that these technologies had tremendous potential to evaluate toxicity and safety. And second, that the path to incorporating them into regular practices was going to take creativity, time, and considerable effort. Now, CAAT has always had a strong interest in how to move alternative science into policy discussions. And when I first got involved, which, as I mentioned, was about 2 decades ago, the majority of these efforts involved working with the business community, some advocacy groups, and also a few policymakers that actually sought out CAAT for its expertise. As a lawyer and scientist, I was fortunate to really work with CAAT to develop a much more aggressive and proactive program, one that really jump-started CAAT into the policy arena. Under Alan's guidance and sponsorship, the policy program was launched around 2006 or 2007, and our goal was to expand CAAT's influence in the policy world. I don't have time today to tell you all about the program's efforts, but I do want to highlight 3 areas that have led to progress. The first thing I want to highlight is our efforts in outreach to attorneys. We redoubled these efforts after the publication in 2007 of the National Academy Report, Toxicity Testing in the 21st Century, a Vision and a Strategy. We made presentations at legal conferences such as the Animal Law Conference. We brought in top scientists affiliated with CAAT, for example, Dr. Dan Krusky, who was also chair of that committee. And we brought them to fora to speak to attorneys about what the future of toxicity testing could look like. We also sponsored a series of 4 international workshops that brought together attorneys and scientists from academia, industry, and advocacy groups. And again, many of these participants were CAAT family. Now, along the way, we were able to break down professional barriers that kept attorneys and scientists from working effectively together. As Professor Frosch, I think, has indicated, one of our goals, and I believe we have been successful in this, and we continue to work on it, is to create allyship among lawyers interested in animal law and scientists who are interested in developing methodologies that don't use animals. These efforts do continue, and in fact, they've expanded so much that I am now privileged to teach a course at Lewis and Clark Law School on laboratory animal law, which I believe is the only law class in the United States dedicated to laboratory animal law. And Lewis and Clark is an international center, so in my class, I have people from all over the world. So our second effort has been basically lobbying directly on Capitol Hill here in the US. And we've been active in engaging federal lawmakers on alternative issues. About 10 years ago, we inaugurated this program, and our first lobbying effort started pretty small. We made 3 or 4 visits to Capitol Hill to elected officials' offices, because we knew that certain officials were interested in animal welfare issues. Since then, though, our work has greatly expanded. In fact, this past year, we met with close to 20 congressional offices to discuss alternatives. Over the past 6 or 7 years, we have also worked with those congressional leaders to directly influence federal legislation. in favor of alternatives. So in addition to increasing our reach through that direct lobbying, we've also sponsored or actively involved in congressional briefings on alternative issues. We were fortunate to recruit Thomas to speak at one of our first briefings, and that was held shortly after he took over as leader of CAAT. And this briefing was actually sparsely attended, no fault to Thomas, who gave one of his consistently excellent talks. But we were new kids on the block at the time, and we hadn't quite gotten the hang of things. Now, I want to just fast forward to this year, where both Thomas and I were invited to speak at a briefing organized by the Jane Goodall Institute in support of a bill that would establish a National Institute of Alternatives at the NIH. Over 300 people attended that virtual briefing. And while I'm not suggesting that it was CAAT's work alone that created this tremendous expansion in interest, I think it's really important to acknowledge that CAAT is seen as one of the leaders in the field, and it is a go-to place now for policymakers who are interested in advancing alternatives and protecting public health. The third thing I want to just briefly touch on is that we do have a certificate program at Hopkins that is meant to foster policy and advocacy. Dr. Goldberg and I first developed a course called Animals in Research, Law, Policy, and Humane Sciences. We taught the course for several years. Dr. Zerlo and Stevens then taught the course, and now it's taught by Dr. Herman, who's really expanded its reach and really brought the course to a new level of excellence. This course is the centerpiece of our certificate program, which is called the Program in Humane Sciences and Toxicology Policy, and it's meant for any student who's interested in learning about the 3 L's alternatives and their policy implications. I'm the faculty sponsor of this program along with Dr. Herman. And you've heard about CAAT's great educational efforts, which are led by Dr. Smirnova, and the certificate program is part of those. Now, as our policy efforts have mushroomed, we've separated the policy program from CAAT to become an independent group within the Department of Environmental Health and Engineering. I like to think of this change as similar to what happens to when a child grows up and eventually moves out of their parents' home. It's a natural evolution, and I really think it's a sign of success. Today, our policy program consists of 2 postdocs here at Hopkins who are lawyers, 2 research assistants who are scientists, and a PhD student who's working on the development of microphysiological systems. And we also have an expert on lobbying in Capitol Hill. Our work is really made a lot easier because of our close ties with the CAAT family and CAAT's strong scientific and technical expertise. We rely on the work of Dr. Maertens, Hogberg, Smirnova, Hartung, and all the other CAAT scholars because it helps us make the case for alternatives and their potential to address the difficult public health problems that the world cares about. Part of our job is to translate these scientific advances for policymakers so we can advance and deploy the lessons of the 3 R's to make the world a healthier place for both human and non-human animals. And that's a job that's made much easier because of CAAT. So thank you for allowing me to be part of CAAT. And I do want to say in closing 2 things. This is the first CAAT program I've been at in 21 years. It's ran late. And if you believe that, I'd also like to tell you that for those of you that have been around the 21 years that I've been around, you haven't changed a bit. Thank you.
Thank you, Paul. Thank you, Pamela. In the interest of time, we will move directly to our final speaker, who will be talking about future prospects, and that is Thomas Hartung. Dr. Hartung is, of course, the director of CAAT, former head of ECVAM. He's organized and co-organized several of the World Congresses on Alternatives and received numerous awards for his work in the alternatives field. One of those awards, we saw him in a photo that Bert presented. I confess I did not recognize him in that photo until Bert mentioned it. A young Thomas. And Thomas, I'll turn the floor over to you, and in the interest of time, I'll give you the last word as well if you want to thank and say goodbye to everyone at the end.
Good. So thank you very much. It's for me a moment of pride to see all of these achievements and also knowing how many things we could not fit into this short period of time. It's also a humbling moment if I see how many things have actually started so long time before I took over. And it is a moment of anger, because sometimes I think, why does it take so long? But it's a good anger. It's the anger which creates energy and lets us do additional things. I would have loved to throw a big party today. The 30-year anniversary is in very good memory for me, because that's where I found my wife. I'm not saying I need a 40th-year anniversary party to find another. I'm very happy. But I take it as a lucky charm for having these decade anniversaries, and we will do something as soon as meetings are possible again. This is a mask which we actually produced, indicating the reason why we are meeting online and not in person. But it has a tagline, and this tagline was the title of an Scientific American article which Alan and myself wrote in 2006, Saving More Animals. And this is, I think, our strong belief. What we are doing is not only for animal welfare, it is first of all for patients and consumers. So this is Alan when he started CAAT, much younger. Silva just calculated he was 41 at the time when this was taking place. And in 2009 I had the privilege to take over late 2008, early 2009. In many aspects, you have seen today that Isaac Newton is always right. We are standing on the shoulders of giants. That's very important. But it is as important to me, it takes a village. And this village is what we call the CAAT family. It is the staff, it's the philanthropic donors, our sponsors, the grants, the board members, all of you who are making possible what we are doing. And I found recently a very nice quote. It says here, if one wants to stand on the shoulders of giants, then one must first climb up the giants' backs. As knowledge accumulates, harder this climb can become. And so this is so much about the big contribution also of our team doing right now. I would like just quickly to show, these are some of our sponsors in more recent time. Unfortunately, some of them I have to name recent sponsors, because for a while already they have not been supporting us. There's a even larger proportion of funding coming from philanthropic bodies, and there's obviously the various European and US funding bodies which have been supporting our work. And if you're missing a logo or have a rich uncle, I always find space on this slide. Please don't hesitate to alert people. I think the important message is people want what we are doing after 40 years of work. 50% of Americans roughly and about 60% of Europeans object to animal testing. And we have seen that policymakers have reacted to this. You see some of the big deadlines which have been set, deadlines in part also to move out of animal testing at a certain time by the EPA and by the European Food Safety Authority. If you give it a look how successful we were in these 40 years, we don't have good data in the US, we all know. And we see from this analysis from earlier last year, there is a slight tendency of going down, but this is not at all what we could consider a big success. And if you look a bit deeper, the report by the European Commission correctly states that we are not including in this figure about 2.8 million animals for breeding, 2 million for tissue donation, 1.3 million for the production of genetically modified organisms, and as we calculated most recently, more than 7 million fetal life forms. All of these fall under the protection of the animal welfare law in Europe, but are not shown in this graph. So whether we are successful at all, or whether the scientific progress and expansion is eating up our progress, is a matter of debate. But there's progress, and I'm very thrilled that one of the areas where really substantial progress was made was pyrogen testing. About 170,000 rabbits were used at peak, and I'm very happy that my own test, to minor extent, but the validation study which I organized and coordinated, in the end, now after 30 years, leads to phasing out rabbit testing, at least in Europe. 30 years. And I put this up to show that progress can be made, but it is terribly slow in our field. At CAAT, we have changed a bit our communication away from ethics, because you will not convince stakeholders on ethical arguments typically. you need other arguments to show. So we are messaging the bad science, the problem of reproducibility and lack of predictivity and its impact on developing drugs, for example. But we're also showing the economical consequences which are resulting, what the money wasted is. But this does not mean, if we're not pushing the ethics primarily, that we're not happy to be still on the right side of ethics and of history as the increasing poll numbers and legislations and overall availability of alternatives show. This is just one example of a paper, Animal Testing and its Alternatives, the most important omics is economics. I believe very much that it is about economic reasons which should make the doors wide open for alternatives, and they can promote creating markets and they can hinder the implementation of new approach methods. I think we are working in a very interesting field between science, politics and public. And in our field especially, the public plays an enormous role. They are influencing, they are through the media, through engagement, they are trying to get influence, especially on policymakers, and this is working. We have been focusing with our science policy programs in the US, as you just heard, but also in Europe on scientific policy advice, because we feel that academia should have a voice here too, and we are successfully promoting these elements. But there's many others. We are working especially with the animal welfare NGOs in this field to use these mechanisms. But it is also social media which are an important catalyst nowadays to make this triangle work and to impact on actual change. For the future, I see that we have actually to move away from communicating science, the typical I'm giving a lecture type of approach, to something which we call here outreach or community engagement. I think we will have to rethink how we engage the public in a way that they take a certain ownership of what is taking place. And I also believe that the fast track for change is through politics. It is not through influencing the scientific processes. We have the tools. One of the tools which has not been mentioned today, but which is more and more influencing my daily work, is the availability of big data and artificial intelligence. And I will not go into the technologies which became available and are changing the face of science and all aspects of life at the moment. But I just would like to say this is something which is new. Because only since 2012, roughly, we have algorithms which are efficient enough and the computers capable of powering them in order to really make use of AI. And this is something which has been really one of the highlights of recent work. Many of you will recall our paper from 2018, 3 years ago already, which was the result of a program of big data, making them available and creating predictive tools. This led to about 500 articles, 2 of them in Nature, 2 of them in Science. So this is certainly one of the most visible programs we have been producing. It has been successfully implemented by Underwriters Laboratories as their cheminformatics toolkit and before as Reach Across, and has been accepted in Australia in 2020. So you can register industrial chemicals, including cosmetic ingredients in Australia, for 5 human hazards with this tool. However, 3 years and nobody on neither side of the Atlantic was willing to validate this tool so far, which I consider a missed opportunity if you consider only that 500,000 to 600,000 animals are used in Europe every year for assays which can be replaced with the current implementation of this tool, and 200 to 300 million euro are spent for these animal tests. And according to our study from 2018, the computer would do the job even better. We are thrilled, however, that the European Union at least funded now some work, which allows us with a large consortium led by Mathieu Winken, OnTox, with about $20 million to promote AI tools based on our technology for new systemic toxicities. But I don't understand why not in the meantime the topical and acute toxicities are taken advantage of. Our work also made me the field chief editor of Frontiers in Artificial Intelligence, so we are really investing into this field in a much broader sense. The other type of technologies which we did not mention as much today are advanced cell cultures. And this is something we have to keep in mind. The cell culture of today is not what it used to be, these pan-fried eggs in a pan hardly any cell contacts, and mainly contact to plastic and cell culture media. We held these 2 workshops, which were published in our journal Altex, by the way, which made a fantastic rise to an impact factor of 6.2 in the meantime, one of the most successful journals in the life sciences, I would say. And the big news of last week, you might hear in our next Catwalk newsletter, the second workshop actually led to a science perspective paper, which was published last Friday. So you can read more about human microphysiological systems in this article with the opinion leaders from our workshop in 2019. We also were thrilled to receive only last week financing from the FDA for a microphysiological system, in this case for our brain organoids, which shall be used for developmental neurotoxicity, a model which already is funded by the EPA, and this funding will go through the Johns Hopkins Center for Excellence in Regulatory Science and Innovation, CERSI. So the FDA gave one of their rare grants to fund method development to our center, a second in our lifetime over the last 10 years. We also are preparing at the moment for one of the big undertakings, like the creation of the World Conference Series. We are preparing now for a microphysiological World Summit Series. And we brought actually 40 organizations together. And you can see here that we are heading towards the first World Summit in this field in New Orleans in next year. And different to the World Conference Series, this shall be annual meetings, with one in 2023 in Europe and one in 2024 on the West Coast of the US. And just last week, we got confirmation of $450,000 support from the NIH, through the National Center for Advancing Translational Sciences, to make this series of conferences possible. So stay tuned. This is an advancement of alternative methods which has impact on all areas of the life sciences. One of the major challenges we are seeing at the moment is the challenge of integrating all these wonderful data. Certainly AI is one way of integrating data, but we also have the systematic reviews, we have the classical risk assessments, which desperately need some innovation, and we have the integrated testing strategies. And I see this as one of the big elements of our work for the near future. Because it is now, from my point of view, mainly a matter of change management. Because we have most tools, but we don't know how to change to them. And the roadblocks are no longer scientific, but they are economic and they are legal. Is an alternative method defendable in a liability suit, for example? And validation, as it is as much as a hurdle as it is enabling. So we have to rethink the validation process because it has an important function to make alternatives happen. And change is more likely to come in the end from politics than from science. So I think it is important that we are not just at the lab bench and promoting science. So let me close with announcing that we are going to start now a discussion on what we call CAAT 3.0. We named CAAT 2.0 what we started a few years into my work here at CAAT, when we saw the transition happening from Alan Goldberg's CAAT to what we are doing here. This is now some 8 years ago. It is a process which took us at the time 2-3 years, and it will take us 2-3 years again to develop a new strategy which is adapting to the challenges of our current time. We are also in this context reshaping our team. At the moment we are recruiting about 5 different people. So if you're interested, let me know. We are happy to recruit from the family. And I would like to close with Charles Darwin. It's not the strongest, but the most adaptable that survive. So let's help CAAT to adapt. And I hope that our goal to have the 50th birthday in Baltimore, and on this occasion a world conference, this is something we are working for at the moment. There's a lot of positive feedback, that at the 50th birthday you see a reshaped CAAT which is fit for the challenges of the next decade and the one to come. Thanks a lot. Okay, so I think we're at the end of our time for our meeting here. Marty has given me the privilege to say goodbye to you. It is unfortunate that we don't have time for any open microphone. I hope you took advantage of the chat. to give your contributions. We opted for an approach which is digestible online because we said more than 3 hours, nobody wants really to listen to all of us. It would have been wonderful now to have a drink and enjoy together. I will postpone this to next time we meet in person. Feel invited. Remind me, we will have a glass when we are coming together again. Thank you very much. I would like to thank all of the speakers. I would like to help those in the background, Camilla, especially organizing these things. Thanks to the team for these last 40 years of enormous work, and thanks to the CAAT family. Goodbye.